Hey everyone, I'm Dr. Stephanie Shuttler, a wildlife biologist who's been in the field for 17 years, and today I have a lot more stories from the field, from my second field season in Gabon, studying elephants. The last time we left off, I was also talking about my first field season. I returned in 2010 to Gabon for a second field season, and this field season, it started off the same as the last one, but it changed a lot, not in terms of the elephants, not in terms of the ecology, but in terms of the management of the field station. There was a new director at the time, and he replaced the field station director who had been there for a long time, and she was amazing with interim directors who had no experience or very little experience in Gabon. And they did not take the wildlife of Gabon seriously. Like, they actually didn't think the elephants were that aggressive. A lot of them had been in South Africa before and just, you know, thought that they're elephants, they'll charge you, and if you stand up to them, then they won't charge you anymore, which does have some truth to it. It's mostly true, but there is a famous elephant biologist, Michael Fay. He was in Luongo National Park with tourists, and there was an elephant, and he was trying to demonstrate this point, that if you stand your ground, they will not charge you, or they will not attack you if they charge you. And what happened is that this elephant actually did attack Michael, and a tusk went like right through, I think it was like this part of his arm, and luckily it missed a really important vein, so he was able to survive, but it was a close call. Now for the last part of my research. And this involved actually taking all the dung samples that I collected. So I had collected, I think, yeah, over 200 fresh dung samples from forest elephants. I put them in tubes with a solution, and you actually had to boil the elephant dung in tubes in a hot bath of water before I put the solution in there, and that was to kill off any pathogens, any potential pathogens, to import them in the United States. So what I brought back to Missouri with me were these tubes of what we would call dung slurries. And I'll put a picture on the show notes, and they had a number on them, and you shake them up, and it has like chunks of like eaten grass by forest elephants and the solution you put it in. So when I got to Missouri, the steps that we had to take was first to extract the DNA. So you want to get rid of all of the plant material, and you want to focus on isolating the elephant DNA. So you have to go through a process that takes 2 days. You actually have to go to a separate extraction room. So again, it's lonely. I went from my lonely field season to the lonely extraction lab. And the reason why you have to do that is because you don't want to contaminate any of the other projects in the room. Or have your samples contaminated. So you had to do this in the extraction room, where you could make sure that your samples were all protected. So 2-day process for these samples, and you can only do like, I think I did like 15 at a time, something like that. So for 200 samples, you know, it takes weeks and months to process all the samples. Then once we had the samples and extracts, they're just in these little tubes, and it's just like it's like halfway full of the extract. And the extract looks just like water. You could never tell it came from an elephant's behind, from their elephant poop. And then we use these extracts to amplify the elephant DNA. So we had questions that we had questions about how elephants were related to each other. And then we had questions about their sex. So were we getting males or females? For the relatedness, we had maternal DNA, so that's mitochondrial DNA, and that will tell you elephants that share the same maternal line. So you get your mitochondrial DNA from your mother always, so that's passed down in that way. And then we had microsatellite DNA, and these are really short, repeating sequences of DNA that have repeating nucleotides. So they have the same pattern of like, so like CA, CA, CA. So your DNA is made up of A's, T's, C's, and G's, and these are thought to mutate faster. And so therefore they are good indicators of relatedness. And the markers are based on the number of the repeated patterns. And we did Pcrs. So basically, polymerase chain reaction, what it does is it amplifies the DNA. It is a series of heating and cooling that mimics cell replication in your cells. That's really what happens in your cells. When this was invented, this revolutionized scientific research. So we were able to create we actually called it a little cocktail that was designed to amplify the elephant DNA that we wanted to. And then afterward, we would run our results on a gel electrophoresis, which is like this gelatin slab, like for real, it looks like gelatin. You put it in this solution and you put a charge in it, and it separates the DNA into pieces because it's hard to move through a gel-like substance. It like slows you down. Imagine swimming in pudding as opposed to swimming in water. So if you're a really big piece, you're gonna get stuck towards the top. You're not gonna move all the way down the gel. But if you're a little piece, you can go fast through that thing. So for some analyses, like looking at the sex of the elephants, that's all we had to do was run it in the gel, and you could tell by the number of bands if it is a male or a female elephant. But everything else we had to send off to the core to get it sequenced. And then we would look at the results of either the actual letters of the DNA sequence for mitochondrial DNA or the numbers of repeats for nucleotide DNA. So that's a lot of detail, but I just wanted to tell you all the different processes that we had to go through. Now, with microsatellite DNA, because we are working with non-invasive samples, so that means we are working with dung samples, hair samples, or feather samples, basically anything that is not tissue, you're not gonna get as good of a source of DNA. So it's possible that your animal could have one kind of DNA signature, but part of it just didn't amplify. So for the microsatellites, to confirm the patterns, we had to run ones that were heterozygous. So for example, if you remember the Punnett squares that you did in high school, where you have brown eyes and blue eyes, you have one allele coming from your mom and one allele coming from your dad. So the same is true for these microsatellites, but we need to confirm those patterns. So if they were, so say for that Punnett square example, you had a blue allele from your mom and a brown allele from your dad, that would be heterozygous. But if you had a blue allele from both, that would be homozygous, they're the same. So for homozygous results, we had to confirm this 3 times. So we had to replicate all that process besides the extraction 3 separate times. Heterozygous, we had to do twice. But the horrible thing about working with non-invasive genetics is that a lot of times it doesn't work because you don't have the best quality of DNA to begin with. I had to do so many repetitions, and depending on the sample too, like sometimes I saw an elephant poop in the field and I knew that sample was from that elephant, and for some reason I couldn't get it to work. So that was really, really frustrating. And I had to play a lot around with the protocols. Sometimes I had to reduce my DNA concentration, sometimes I had to increase it, but basically a lot of time spent in the lab. And I did have to discard some samples that I really cared about. I couldn't include them in my study, which was heartbreaking because I spent so much time collecting those samples, but that's just the nature of non-invasive genetics. So thank you guys. Cut that. For more on my research, make sure you watch the previous videos. If you like learning about animals and science, subscribe to my channel to get a lot more information about the kind of research that I do, as well as research out there in general. Bye.